Alright, so we left off doing PC1 and kind of just finished off at review, and I think I stopped when I told you how to identify whether you have a 5' UTR or not, or if you have a 3' UTR or not. So the very first thing you need to do is open up a blast P. I'm going to close your mind for now. And run blast P. Alright. While that happens, let's go look at the questions. Just read through your analysis questions and voila. So you're just gonna click on this blue bar. I mean, you don't have to wait for all of this to load. You can just click on it as soon as it appears. You can close this tab if you want. It's gonna take you to a page like this. So to tell the function of your, so the very first question is gonna ask for the function of your protein and the name of your protein, and that's gonna be in this tab right here. The very first one, the specific hit. You're gonna read through this little speech bubble. Well, it's not a speech bubble, it's like a little pop-up bubble. Whatever it's called. You're gonna read through it, and it's gonna tell you the function of your protein. Now, it's okay if you don't understand it, read through it a couple times. I mean, I don't expect any of you to know what thyrodoxin is or what any of, I'm pretty sure you don't know what this is. But just read through it, and it's gonna tell you what the function of the protein is, summarize it in the best way you can, and move on. The worst that can happen is that you get it wrong, and the PC is bounced back, and they just tell you to fix it with this, this, and this, and they tell you exactly how to fix it. So you're fine. Number 2, was there a similar protein found in humans? So I'm gonna open this link in new tab, but you're gonna click on the dark blue, and it's gonna take you to a page like this. That speech bubble thing that I have a confusion for naming. That thing is this right here. This little thing, this paragraph. So you can read it over there, or you can read it over here. It doesn't really matter. But you're gonna scroll all the way down, and this protein right here, CD03419, is this right here, 1-KTE. So you're gonna click on it, and here is a little page for that protein. Oh, sorry. You're gonna write down, oh wait, sorry, I apologize. You're not gonna click on that. Sorry. I'm so sorry. This is your, the closest match, and this is this protein right here. What you're gonna do is click on all of these to see if any of them are Homo sapiens or humans. Because all of those proteins, that list of proteins, down there that I was going through is, they're similar to the protein you have. So lucky for us, the very first one was Homo sapiens, so you could just write down yes. And what exactly does this tell you about the protein? Well, I'm sure you've all seen that little evolution tree chart thing, and in that we see that humans and plants are very far apart, that the branch, their common ancestor, is way, way, way back. So it kind of says something that this protein survived all of those mutations, all of that evolution, and it's still similar. That's gotta be a pretty important protein to have survived all of that. So you can see that the protein is highly conserved and it's important and all that good stuff. Number 3, does your protein contain domains? So again, you go back to that little blurb, speech bubble, and usually stuff about the domains is in the end, or you can, it's just read the thing again, it's gonna tell you about domains. Excuse me. Glutarodoxin family, GRX human class 1 and 2, like subfamily, composed of proteins similar to GRX's, approximately 10, I don't know what this unit is, kilo, I'll figure out later. The proteins containing wait is composed of proteins similar to the human glutaredoxins and composed of proteins containing GRX, or GRX-like domain. So it says that it is composed of GRX-like domains. So you can say yes, there is a domain. Also, in the end, you don't have to read through all of this. Well, I mean, you do when you're doing other PCs, but I'm gonna help you out with this one. So in the end, it talks about hold on. Different classes are known, including human GRX1 and 2, which are members of this subfamily. Also included in this subfamily are N-terminal GRX domains of proteins similar to human thyrodoxin. So, yes, domains clearly do exist, but how do you tell the function of these domains? Well, here are all of those different domains. So you're going to look through these, and if it doesn't tell you the function, you're going to look at the last superfamily. Fortunately, the thyrodoxin superfamily has a little blurb. You can read through this, and turns out the function of thyrodoxin and glutaredoxin is pretty much the same, except, with different target proteins or target substrates. And that's exactly what I put down. On to number 4. Can you find a 3-dimensional structure for your protein? Well, there's a picture here, and that's a 3D structure, so yes, I'm pretty sure I can find a 3-dimensional structure of my protein. So you put down yes, and like I said, this protein right here is this right here, KTE. You're going to click it, and you're going to look at what organism it's from. Zooscrofa, I think. That's a pig thing. I remember looking it up, and I thought it was a buffalo, but that's not a buffalo. And the only other animal I remember looking up is a pig, like a wild pig. So yeah, I don't know. Anyway, so yes, there's a 3D structure found, and it's a Zooscrofa, and you write in the accession number. And then it asks you how many alpha helices are in there and how many beta strands. We can use this. Yes, it's the same thing. But actually, I like the colors of the other one. I'm sorry. I apologize for my indecisiveness. Alright. Okay, so the green spirally things are alpha helices, and the yellow flat things are beta strands. So you're gonna just count them up and write it down. 5 beta sheets for beta strands. Alpha helices. 5 alpha helices and 4 beta sheets, and they are both parallel and anti-parallel. Why did I see that? Because the alpha helices are not parallel and the beta strands appear to be parallel. Yeah, so they're both parallel and anti-parallel. For number 6, you're going to put NA. Well, all of your PCs are going to be NA, but what that question is asking is comparing your estimated sizes with the actual size of your insert. So this is my own clone. This is what I've analyzed and published. So when you're cropping ends, right, you take very specific base pairs. And with this, you determine the exact size of your protein, and what was I, 314 base pairs. And then you compare it to your PCR and RDG estimates. And my PCR was much further than my RDG estimate, and that again shows you that, RDG is a better, more reliable estimate, more reliable test for your estimation, and that's all you have to do for your number 6. But for PCs, like I said, you're going to put NA. Where is PC1? There it is. So, okay. Last but not the least, definitely not the least, is number 7. Number 7 asks you what else you could do with this protein. Now, this is the one question that most people have trouble with. What else can you do with proteins? So, the most common answer for everything should be crystallize the protein. For all of your proteins, they haven't been found before, and they do not have their own 3D structures. This 3D structure is from that protein, is from a protein found in that little pig thing. It's not from Landotia punctata. So what you could say is that you could crystallize this protein and determine its actual structure. Another thing is you can knock out the gene that codes for this protein and see what it does to the cell. Does it kill the cell? Does it like have certain effects on the cell? Um, you could do a ton of different experiments. I also said that you could compare the structure of your protein with glutaredoxin from Homo sapiens, and then determine differences in structure and function. And number 7 is just basically you sciencing out, like bring out your inner scientist. Talk about all of those like sciencey tests. If you don't know any, Google them. What can you do with a non-crystallized protein? I don't know. Google. And the last step is going to be submit. Over here, you're going to write down the name of your protein, and you're going to put down coding, non-coding, RNA or mitochondrial for all of your PCs. No, I lied, not for all of your PCs. For PC1, it's going to be coding. A lot of people asked me what BlastX wasn't, well, not a lot. A couple people asked me that BlastX is not working in PC2, so we're going to run through that real quick. BlastX. So we have to take into account what exactly is BlastX doing. BlastX takes your protein sequence and runs it against, no, I'm sorry, your nucleotide sequence. Hold on, I need to load Toolbox to show you guys. Toolbox and CBI. Alright. What? Why you gotta do this? Oh, sorry, it's not on CBI, it's on WSSP. I'm a funny child. Resources. 2 Alright. So, what does BlastX do? It takes this nucleotide sequence you have, and it splits it up into the 3 different frames possible. So, what exactly is this doing? Because, hold on. Is the retrieve sequence still? Yeah, okay. So, because a codon is 3 nucleotides, you can, it starts out, it can start out in 3 different positions. It can start out with the A, so the first codon is ACG, and then the next one is AAA. Excuse me, so the next one is AAA. Or it can start out at C, so that the first codon is CGA. This is frame 2 now. ACG was frame 1, CGA is frame 2. The next codon is AAA, on and on and on. Or it can start out with GAA. So that this is frame 3, right? But it can't have a frame 4 because this AAA is part of frame 1. Because remember, these 3 were one codon, and these 3 would be the next codon. So the most you can have is 3 frames. What BlastX does is it turns them into protein sequences, and it takes all of these, it takes this nucleotide sequence and splits it up into all 3 different frames. And it runs all 3 frames in the database against, all 3 frames against all of the proteins found in the database. That's why in the BlastX, it gives you the little frame number at the end. So now if your BlastX doesn't work, what does it say? Were there just no matches? Is it possible that you just found a protein, and nobody has ever found this protein, and in any organism there's no matches anywhere in the entire database? That's highly unlikely. But it is possible that your sequence just doesn't code for a protein. You just don't have anything that codes for a protein. So you leave your BlastX empty because nothing shows up in your BlastX. Your define ORF, you just put down no frames because it's not coding for anything. BlastP is out of question because if it's not coding for anything, there's no protein. If there's no protein, what are you going to BLAST in the database. And in the analysis portion of it, it gives you 3 questions. The first one, it asks you for the actual insert size. Again, you're going to put down NA because it doesn't apply here. And then it's going to ask you what experiments would you do next. I put down that the sequence is not encoding, and as a possible reason, I put down that it could be from an intron, not an exon. So, what was I saying? Intron, sorry. So, if it's from an intron, obviously it's not going to code for a protein. And the third question is going to ask you to run a BLAST2 sequence. How do you do this? You're going to go onto Google. Oh, I'm so sorry. That was awkward. BLAST2 sequence. BLAST2 sequences. And then you're gonna wait a minute. You're gonna take this and you're going to run it against itself. Oh, what? Why? Alright, let's go. Line 2 or more sequences. Does that happen in BLASTX? I'm just a little curious. I'm gonna be a curious person. Ooh. This is not time for experimentation. I'm so sorry. I'll do this next time. So you're gonna make sure you're in BLAST N. You're gonna click align 2 or more sequences, and you're gonna paste it in both and just blast. Alright. Where'd it go? Okay, I'm gonna close Toolbox. It's going to ask you for, oh, sorry, if there's any repetition. Repeated sequences on the DNA. If there are repeats, why do you think they are present? Okay, to be honest, I read over this. I don't know why I put this. They are particularly important and highly conserved in the cDNA. In the cDNA. I don't want to revise and submit it again. But whatever. I'll ignore it. We can ask Dr. Lawlin about that. But for now, yes, there are repeated sequences. Repeated sequences is this bit, this gray bit right here. Right here. That's repeated sequences. Yep. Yeah, I don't know what else to, like, I don't know why I put this. But anyway, it's gonna tell you that, and that's that. In the submission, you're going to put down it's non-coding. You don't have to write anything down here. It tells you that these errors pop up, but it also gives you the option to submit with the errors, and I would submit with the errors. Yeah, that's it. That's the end of that. The end. Alright, have